To answer the two questions that we have today, we will first talk about the steps required in DNA replication and then pause briefly in that process to determine how one can identify a leading strand from a lagging strand. To start off, we begin with a double strand piece of DNA. One end of this will be 5' and another 3', and its corresponding pair will be the opposite as we pair nucleotides. After this step, helicase comes in and splits this to create a replication fork. Single-stranded binding proteins then come and join the areas over here to keep the replication fork open and relieve tension. Primase will then come in and add DNA primers to give DNA polymerase a place to start and continue the rest of the process. At this point, we can talk about how one can determine a leading strand from a lagging strand. It is important to note that templates can only be read from 3' to 5', and that our leading strand must be continuous, where our lagging strand will be discontinuous. In order for our leading strand to be continuous, we must find an area that begins with 3', because this is the direction that template is read, and follows another rule, which is that it must go in the direction that the replication fork opens. It's also important to note at this point that at any given point, a nucleotide can have both a 5' end and a 3' end. And therefore, this allows us to put a 3' end or a 5' end arbitrarily anywhere in the replication fork. So, in order to meet both our requirements, we can add a 3' end here and a 5' end here, and the opposite here. So we must now fill our requirements to determine a leading strand. We find a 3' end that goes in the direction of the replication fork. This only leaves us with two options, this upper left segment and this bottom right segment. These are both now leading strands. This would make these two remaining strands our lagging strands. At this point, we have primase, which has added the primers to our replication fork, and DNA polymerase has come in and added the complementary nucleotides to each area. We will then have nuclease will then come in and remove these DNA primers. And this will, as a result, leave gaps these gaps in the nucleotide sequence. Nuclease will degrade the primers and it leaves a gap. Another set of DNA polymerase will then come in and fill out these areas to make sure that it's a continuous strand. The only difference between this process and the process of the lagging strand is that because the lagging strand is not able to be read continuously because it's being read from 5' to 3', it creates what's known as Okazaki fragments. And so as DNA polymerase and primers read the nucleotide sequence, it creates little loops known as Okazaki fragments, which then later on must be combined together. This is the job of ligase. Ligase will then come in and bind together the nucleotide sequences known as Okazaki fragments to create a continuous nucleotide sequence.